or just the drug? I mean, is it dose dependent or?
Yeah, usually it's dose dependent, and this is a very good comment and question, because I did not go into detail, because we do not see it very often in the clinic. But we know that this so-called pseudo-allergic reaction, meaning they look like an allergy, but it's not IgE mediated, that these really are mediated by complement. And this we know best from contrast media, for example, but also from some lipid carriers of drugs, but also from some drugs, as you mentioned. First, of course, we have to make clear, is it really complement, or is it direct interaction with the mast cell? I don't know. Did you check this drug?
Yeah, we are studying it at the moment. We are
Yes.
collecting samples. We'd like to measure some complement.
Yes, yeah. So it would be very
like
interesting
CC.
to see if those patients do have a high activated complement system,
Yeah, I think.
or if they have a normal one. Yeah.
Yeah.
And then I would expect, as we know it from the other drugs, that it is really dose dependent, the activation. But for a certain threshold, of course, we may have the same symptoms.
The other question is, the first question. You get this with amphetamine, just amphetamine? No, we do not observe this with. It is the liposome.
Could it be the liposome?
Yes.
Because the liposome is a common vehicle now for
Yes.
drug delivery.
So from other liposomes, we know that they're really
Yes.
activating
It's not,
complement.
it's not the amphetamine, it is
Yeah.
the liposome
Yes.
in the amphetamine.
So that's the generalized problem of
It's
this.
true,
Yeah.
absolutely.
And this depends on the charge of the lipids, which you have integrated in the liposome. We are currently in a different project looking for different assembly of liposomes, doing from neutral lipids and from charged lipids. Most lipids are usually positively charged, and they are very much activating complement. But as soon as you get them neutralized, by any means, or you use some neutral lipids, then you may reduce the complement activation capacity.
Yes, so the second question is about hypocomplementemia. In case of renal disease, like
Vasculitis and so on.
is it due to the outcome of the disease? I mean, the loss of these complement proteins in the urine, or it is a risk factor?
I would say both. Hypocomplementemic vasculitis, for example, or in the critic situation, is a combination of loss of protein by proteinuria and consumption by activation. And the second, the consumption due to activation, this is a risk factor. If you have a strong local complement activation, this triggers the disease progress.
Is there any way to intervene to modify the disease progression, for example?
Yes, actually, what I mentioned in this disease called atypical HUS, which is also a severe local renal disease, where we have success with the antibody against C5 called eculizumab. This is not approved yet for all kinds of diseases, so it's not available, and so it's an orphan drug, and it's just currently tested in a multi-center study. But if this, what I expect, will have a very positive outcome, then we may have to think on other new forms of nephritis, where also complement is severely activated, contributing to the disease progress. And I could imagine that then it would also be probably a drug of choice.
Thank you very much for this nice presentation. In malaria point of view, we know that antibody alone does not opsonize or kill the parasite.
Malaria?
Yes.
Yeah.
So there is especially when we talk about functionality of antibodies, we call it broad inhibition antibodies. There is now a new trend is thinking that the complement system is very important to enhance this, the function of antibodies. But do you have an idea about how the complements would interact in vitro assays? Do you have any idea about any kind of in vitro assays where we can put more, add complement antibody and see effect on anti-parasitic effect?
Of course, you can do in ex vivo experiments or in vitro experiments where you have to separate everything, the pathogen and all the infected cells, and then, for example, serum as a source of complement. And then you can see also kinetically if you are able to kill either the parasite or the infected cells. This is a general approach I would recommend. I have to admit that I haven't been working with malaria with respect to complement. I know that malaria, that there's an interaction with some complement receptors on the cell, yeah, of the malaria-derived protein, but this is everything. And for a while, many years ago, we thought we could work a little bit more on the recognition of the role of complement in malaria, but we stopped this because we didn't have enough access in Germany to all this. Yeah, but this is a very interesting feature that now you think that it may be that specific antibodies may be able to block the let's say the inhibitor growth. Yeah. But I think you can only do it. You can do it in vitro assays by taking complement and adding either to the different forms of the malaria or the infected cells, and then you can use this assay to look is this system activated or not, or you can take out the cells and look with fluorescence analysis, are complement proteins bound or not bound. So this would be a general approach, and maybe this would fit to the current notion what is how, or if at all, complement is contributing to the success of certain antibodies. Yes?
Pathway or lactin mannose pathway. In classical pathway, we require immunocomplex formations. In alternative pathway, we require cell surface antigens. And in mannose lactin pathway, we require lactin and mannose. In case of BNH, which is one